just wait one more minute. People transition time. Cool. So why don't we get started? Welcome, everyone. Today we have Daniel Barabási, who is currently a Schmidt Fellow at the Broad Institute at MIT and recently finished his PhD at Harvard. And he's going to talk to us about physical network constraints and the brain's connectome. So Daniel, thanks for coming, and take it away.
All right, thank you. So I guess I want to preempt this talk by saying that this is a work that we hope to have had pre-printed by today, but we haven't, which is actually both kind of a little bit nerve-wracking because it's the first time giving this talk and it's a little bit more raw in that sense. But it's also a great opportunity to provide feedback. So I don't know what the setup is on their end because I see a physical room, but if there are any questions, if you could signal me in some way, then I'm really happy to answer them during the talk as well, in order to get more feedback before submission. So generally, the idea of the talk is that we had this opportunity in the last year to look at a diverse number of connectomes or brain wiring diagrams, where the change has been that we've been able to look at this at the cellular level. And there's a number, we have this first opportunity to do comparative connectomics, to do like this comparative studies on this. And we have some exciting results from a network science sense. And also it's somewhat of a call to action to say that there's lots more to be done on this front. So I just want to begin with some concepts of what a connectome is and how it fits in with the rest of the omes. Over the last maybe 30, 40 years, there's been this idea that you are your genome or you are your DNA sequence. But this has been a little bit refuted in the sense that every cell in our body roughly has the same DNA, but we have very different cells. We limit confuse, the cells of our finger with the cells of our brain. So the idea that you are your genome is a very first order approximation of who we are. So over the last maybe 20 years, we've been looking more at things like transcriptome and proteome and epigenome, which is more about how we use these genes, what sort of proteins are actually being expressed, and how each cell utilizes the genetic material differently. And this has been, in certain ways, a massive success in both our ability to develop new drugs and to both understand and to address diseases. And also, there's been many of these cell censuses or brain censuses where you've looked at all the different cell types. But when you think about who you really are, you don't really think about what genes am I expressing right now or what sort of proteins are circulating within my body. But a better idea of this is maybe related to this video, right? Because a lot of who we think we are is more about what we do and how we interact with the world. And a good example of this is this cuckoo bird, which has recently hatched, right? And it had no ability to learn within its life, yet it executes a very complex motor program. And I love this part because it's knocking out these eggs, and here's a part where it almost falls out itself, but it still stays within the nest, right? So what's going on here is through development, through the use of the genome, the transcriptome, proteome, and so forth, a certain brain wiring has developed within this animal that allows it to execute these behaviors and survive within the world, right? So rather than being our genome and all these things, we are more our connectomes than anything else. And the reason that we haven't focused on this so much is because the access to these data sets has been very difficult. But I guess in recent years, as I'm about to show, we've had more and more access, which will allow us to start answering questions like, where in development does a certain connection come from? How does a certain synapse or connection within the brain encode a memory or our personality? And how does it give rise to unwanted or harmful tendencies? So we have to start developing the methods, the analysis methods that when we have a network of the brain that allows us to get at these connections between structure and function. And just to start us down this road, given that this is a network science seminar and not a neuroscience one, just want to touch briefly on what a neuron is and why they look so weird. So most of the cells, maybe in a biology course that you looked at, are just kind of round objects with a nucleus at the center that stores the DNA. And neurons still have this, but they also have all these other branches that come off of it that like the dendrites where it receives connections from, and an axon where it sends out connections from its body. And these two different parts of the neuron define its kind of output and input, or input and output sequence. And on top of it, it's a physical instantiation of computation. As it receives information or activity from its dendrites, it integrates them in the cell body and outputs them on the axon. And there's all sorts of physical rules that apply through this, right? And the issue is that neurons don't all look like this. There's a huge diversity in the types of neurons there are. One of the most famous, which I'll come back to at the end of the talk, is this Purkinje cell, which is within the cerebellum, where you have, again, the soma, as I was showing here, the cell body. You have this huge dendritic structure. It receives a lot of different input. And then it has this output axon where it sends out information. And again, you could see other diversity where these smaller cells or basket cells, for instance, that have different connectivity profiles within the system. So from a network science standpoint, this sort of representation where we could say that every neuron within the brain is a node within our network, and the connections between them, or the synapses that they make, are links. You could use this representation, and we'll start with this representation, but there's an added level of complexity that we've recently gotten access to that we want to consider in terms of the locations of the input, of where these cells are receiving information and where they're outputting them, as well as the number or the weighted strength of these cells. So that's where we're going. So in this way, the brain connectome may be one of the last terra incognitas for network science, and that it was almost 150 years ago that people started to, especially Ramon Call, who has these beautiful drawings, which he has a book on them, which I highly recommend, that he started characterizing staining brains and imaging them, and saw that there's this kind of network structure of individual nodes as neurons. But it was only 100 years later that we had the first cellular level map of really any organism. And in this case, in the C. elegans, which has 300 neurons. And these 300 neurons, he was able to map the connectivity between them using a technique called electron microscopy. So this was in 1986. And since then, up until basically last year, we've made very little progress on this front. But last year, the Connectome data availability exploded massively. So it showed that in 1986, there was the mapping of the C. elegans with 300 neurons in the reconstruction. I'll make this differentiation as to how many neurons are in the whole organism versus how many are in the reconstruction. So this is how much the experiments were able to reconstruct out of the full amount, which will be more important, we'll see in the mouse and human. So in 2020, we saw that there was half of a fruit fly brain and a part of a zebrafish brain. In 2023, we saw a full fly larva being reconstructed, or the full brain of a fly larva being reconstructed. But then in 2024, we had the kind of the hindbrain, which is responsible for motor programs in the fly, the full fly brain, so the kind of the frontal regions, and parts of mouse and human being reconstructed. So in this way, for the first time, we had, in certain cases, a complete picture of multiple organisms. And in other ways, we had very nice representative samples of the mouse and human. This allowed us for the first time to perform somewhat a comparative connectomics approach and start to compare these networks and see if there's any similarities or differences in the structures of these networks. And just as a, let me see if I can switch this to video sharing. Great. So the way that connectomics works, or how these data sets are actually collected, is they utilize electron microscopy to reconstruct it. And what this means is that the tissue of the brain is fixed in this fashion, and is then imaged through an electron microscope and continually cut in order to make these layers of to get the full 3D volume. And then what first humans, but then now mostly algorithms do, is that they go through this, and then given this grayscale image, to identify that this is a neuron over here. And you have to go through layer by layer, as this video is showing, and identify all the cells in it. But as you do this, you can add up all the stacks, and you can find individual neurons that are kind of projecting through this volume, and you can reconstruct not just their morphology, but as we're seeing over here, their synapses. So what these data sets allow for is it both provides this neuron shape, as I was showing over here. You see the shape of some of these cells, as well as the locations of their connections, like that these two cells are making a synapse specifically over here, right? So it provides an added level of information past just the degree sequence of these neurons. So given this information, we can start to look at first the degree and also various other network metrics and start to understand where these connections come from. Great. So the first question is, what would our expectation be? Well, network science has a very long history of starting to understand what the degree distribution or the distribution of connectivity profiles, how many partner cells are making within the brain, would be. So a lot of these expectations would be first related to random wiring, where the Erdős and Rényi in the 1960s, they would predict this Poisson distribution of a random network, or you might have a growing network, like the brain is a growing network. It has to develop and has to make connections, from which you might expect exponential connectivity distribution. On the other hand, you might have growth in preferential attachment. Maybe cells within the brain are connecting to other superconnectors, and maybe there's a way that during the growth process that this is established, in which case we'd be looking for a scale-free network. And real networks are often characterized by non-trivial combinations of these elements, so that's also something that we'll have to think about as we go down this analysis route. And in the past, people have found that the brain networks or different brain networks align with different types of degree distributions. So in 2000, from Gene Stanley's lab, there was a C. elegans characterization that claimed that it looks like a randomly growing network, so which is often considered model A. Whereas this is a very different type of data distribution and a very different scale, but at the human interareal connectivity level, people have claimed that there's a scale-free distribution in connectivities there. So there's already been this debate as to where it falls into, and it falls into very previously well-known characterizations. So when we looked at no degree across all of these different organisms, first we considered each neuron to be a node, and the degree was the number of distinct other neurons or other cells within the brain that it made connections with or synapsed with. We found a very interesting but repeating distribution across all of them. And here what we're doing is we're plotting on a log bin, so on a log x-axis, so the log of the number of partners that cells have, the distribution or the probability of that value, right? And we found that there's this repeating structure across all the different organisms. And just to check that this was true, we rescaled, where we took the mean of these values, and we see that the mean is obviously different between them because there's much more cells in flies, for instance, and mice than there are in C. elegans. So of course you're going to expect that there's more neurons and bigger neurons, and therefore the possibility of higher connectivity within these larger organisms. And we also took the standard deviation, and we divided this out of each of the organisms. And when we do this, we find that all these distributions, independent of which animal they come from, fall onto the same, seemingly the same plot. And it does seem that all these distributions, when rescaled, are well fit by a normal distribution on a log x-axis. So this was very interesting to us, but we want to dig a little bit deeper because we had access to weighted information and synapse number within these cells, and we want to see whether that gave us a more complete picture. So the next thing that we looked at is node strength. So we looked at the number of synapses that each cell made in total. So if it made 10 with one, 7 with another, and 3 with the third, 3 with the third, then the total number of synapses or total weight of each node or neuron within the brain would be, or this neuron would be 20, right? And again, we saw the same distribution popping up. And we did the same exercise. We rescaled each of these data sets, and we found that again, upon rescaling, they fall into the same distribution, perhaps even more nicely, and and it again was well fit by a normal distribution on a log axis. So this led us to conclude that a normal distribution on a log axis corresponds to a log normal distribution, and we were kind of surprised to find that despite the fact that you might expect, or we were expecting this to be either an exponential or a power law distribution, we found that instead a log normal seemed to, at least by eye first glance, be providing a good fit. But we were a little bit worried, and just for a slight detour, we do have to talk about how log normals and fat tails are considered, right? So one of the things that both in statistical physics and network science we love looking at is these fat tail distributions. And there's many distributions that are a good fit to them. And there's been this big debate as to whether a certain distribution is log normal or power law. That's been in the literature and it's been at conferences. So we want to take a slight detour. So we just want to note that a log normal distribution is mainly defined by the scale parameter mu, as well as shape parameter, which is the standard deviation of the distribution sigma, right? And depending on the value of sigma, you'll get very different looking distributions, right? So here, we're plotting on a log axis, and then we take a fixed mu, but we change the sigma or the standard deviation of this. And we note that for a small sigma, for instance, for all of these, the peak of the distribution will be at mu minus sigma squared. And when you have a small sigma, you'll see a narrow peak, right? And you'll see this very characteristic log normal or normal looking structure on the log axis, which has a fat tail both in the left and in the right, so towards the small and towards the large values, right? What's interesting about this is that you could fit a power law to the right hand side of this, and it'll come up with a very good result. You could do a very good fit, and sometimes for good reason, but sometimes for just because people chose to ignore it. You'll ignore the left hand side, saying that it might be noise, or it might be it might just be like a data quality issue, right? And this has been one of the reasons that perhaps, either that we might have seen log normal distributions in the past, but fit a power law to them. At the other end of this, when you have a large sigma, the distribution gets pushed here all the way to the left, and you only see this right-hand decaying part of the distribution, right? And in this case, you only see the long tail, which you might, again, be think to just fit a power law to it because you don't see the kind of extension to the left-hand side of the log normal distribution. So network science, so one of the reasons, again, that we turn to power laws often in the past is that we pride ourselves on our ability to understand the mechanisms and to analytically predict why we see a specific degree distribution. And in this context, there hasn't been a theoretical justification for log normal degree distribution. Again, a power law has a good story to it as to why a certain growth process or a certain developmental process might lead to a power law distribution. So when picking between, let's say, a power law and a log normal, we might choose the power law because we can understand where that network is coming from and build a better story with it. And so thus far, log normals, fitting these log normals has mostly remained a statistical exercise. And just to push that statistical exercise through, we do find indeed that to all these data sets, the log normal distribution offered the best fit. So the power law was quite poor in fitting all these data sets. Log normal was the best for the degree distribution. And for the node strength, we find that there's certain cases where exponential might be a better fit, but overall across all data sets, the log normal is the best fit. So we basically got to this point where we're saying like, okay, the log normal seems to be the best fit, but we have no understanding of why this is actually, why a network can end up being a log normal distribution. There's a few models that did lead to a log normal distribution that people have explored in the past in citation networks or social networks. But in terms of the brain, the developmental process is so unique and so different and also very pre-programmed that we didn't quite understand where this could be coming from. So in order to kind of get at this at a deeper level, we wanted to turn to the physicality of the system and see if the physical nature of the brain offers an added information for us that we can turn to. So in this context, as I sort of set up previously, up until now, we've been mostly looking at neurons within the brain as something like this, where we have a number of nodes and we have the strengths of connections between them. However, the connectome is a physical network, and these neurons, as we see over here, are physical objects that serve as links. The physicality of the system sets up who you can connect to and how strongly you're connecting to it, past even the number of synapses that you're making. So we see that these connections between the green neuron, which is this, again, this long extended object, and the blue neuron, they're being made in this entangled mesh over here. And the neurons have to be close to each other. They have to be physically next to each other in order to make these connections, in order to have this availability. And also, maybe the distance from the soma and the density of connections is very important in terms of how strong those connections actually are. So looking at this physicality allows us to kind of dig deeper into how these connections are being made and why the degree distributions are as we observe them to be. So, and on top of this, all of these neurons are rare characterized. So these are 3 cells from the fly brain, where you can look at the length of them, the density of connections, and there's different neurotransmitters. So this defines a kind of a positive or negative effect onto other cells. And they all have certain cell types that we can also utilize within our analysis. So why does physicality matter? Well, it allows us to come up with these hypotheses as to how the physical system is linked to the observed degrees which we had been focusing on thus far. So the first hypothesis that we had, that the longer a neuron is, the more opportunities it has to form synapses with other neurons, right? This makes sense. If you have the more available physical space available for connections, the more connections you'll make. So just to state this in a mathematical way, we can say that the synapse number that we observe is proportional to the density of connections to the synaptic density, as well as the length of neurons. And in looking at the data, we find that this is true, right? So we find that there's a nice correlation between the length of neurons and the synapses. But since L and rho are these covariates for the number of synapses you make, and the number of synapses you make determines your possible degree, we need to understand what the nature of the length and this density distribution are, such that they lead to a log normal synapse distribution. And in measuring this, we again saw a log normal distribution across all of these, which I'll kind of better show through this renormalization, that both the neuron length and the synapse density seem to also follow a log normal distribution within all of these data sets. I'd like to take a moment here to talk about this data, the fact that we don't have data for C. elegans. One of the major parts that one of the major avenues of this research was actually going through downloading the data and cleaning it. And despite the fact that there's standardized approaches for releasing connectomics data, there's still a lot of caveats as to how this analysis and what cells you could actually trust. This is a process that we're hoping to release as part of even the preprint, so that you can go and kind of look at our clean versions and people can kind of debate the cleanliness of the data, what are the good cells to use and which ones are not. And for C. elegans specifically, we just didn't have a data set for which we had both neuron length and synapse locations within the same data set. So we knew the synapse number of these cells, but we didn't know where along the neuron the synapses were. And we're actually using a local density metric here and not a global density metric. So that's why we have no data here. But this is also something I'm happy to discuss as to how you get access to data sets like we're using within here. So return to the story, we find that this length and density distribution are also log normally distributed. So a brief primer on log normals is the fact that they're, again, defined by this mu and sigma parameter. And if you take 2 distributions that are log normally distributed, then their product will also be log normal, and you could derive the expected value of the outcoming log normal from within the system. right? So you could say that the resultant distribution has a certain log normal that's dependent on the parameters of the two input functions. As we'll get to a little bit later in the talk, this is also true when you take the power of it, of a log normal, and you'll get a different, you'll still get a log normal out of that as well, just with a different parameterization dependent on the initial input log normal. So the reason that this is important is because we found that the length distribution and the density distribution of neurons is log normally distributed. Thus, the synapse distribution we predict will also follow a log normal, where these parameters will be dependent on the input parameters. Specifically, these are the values that we predict, and we find that there's a very nice correspondence between the predicted mu of the synapse distribution and sorry, the measured one and the predicted one. Across all these animals, there's a very nice fit between them. And the same for the sigma values between these. So given this prediction that's only dependent on the length and the density distribution, we get a very nice prediction for the number of synapses that cells should be making within the brain. And just to show this more explicitly, here are 3 different data sets from the zebrafish, from the fly, and from the mouse. And the curves are not fits, but they're the analytically predicted synapse distribution based off of the length and the density distributions. And we find that there's a nice correspondence between the empirical data and the prediction that we observe. So this was one of the first cases where there's a nice match between the kind of the physicality of the system and the observations that we have. And just to get to a second one, we looked at the degree distribution, and we thought of the fact that a neuron's ability to acquire synapses would be sublinear, because when you think about it, if you have 10 synapses, right, if you have a neuron with 10 synapses, then there's 10 total partners that it could have, right? So each of the synapses could be with one partner, canonically. And but often what you'll do is you'll place multiple synapses onto a single partner in order to have a stronger influence over them than perhaps over other partners. So your total degree is going to be thus not equal to your total number of synapses, but slightly lower scaling than that, right? And indeed, we find this is true, that the synapse number and the degree have this really nice correlation across all these data sets where they're all a sub-1 correlation. And what I showed you earlier is that even within this formalism, if the synapse distribution is log normally distributed, then we predict that the degree distribution will also be log normally distributed given the underlying synapse distribution. And this is the values that, or this is how the relation should occur. And again, there's a nice fit across all data sets between this prediction from the physicality and from the measured values. And finally, again, what I'm showing is not these fits, but the predicted degree based off of I think this should be S's, but based off of the synapse distribution and these fit parameters over here. So there's a nice correspondence between the prediction and the synapse values. So what we've been able to do is we've been able to utilize the physicality of the system to step our way up from the neuron lengths and the neuron degrees, which we find to be log normal. And we can get from there to the synapse values, and we can get from the synapse values to the degrees. So the only thing that's missing is how you get log normally distributed lengths. We have a model for that, but the model is it takes a long time to go through, so I did not include it, but perhaps through questions we can get to. But basically, we utilize a branching tree, and given that neurons are branching trees, and we find that a Galton-Watson process gets you the neuron tree structure, and that therefore, through a multiplicative process, you get a log normal length distribution. And we have some nice theory for that. That'll both be in the paper and in the SI, and I could touch on a little bit depending on interest. But I want to also get to why this is interesting from a network science standpoint and what more we can do in the future to analyze data sets like these. So a final kind of interesting tidbit for those who are a little bit more neuroscientifically inclined is that the log normal distributions extend to cell types, right? So far we've been looking at the brain level, or doing a brain level analysis where we look at every single neuron within the brain, but there's a lot of diversity in the sorts of neurons that you actually see. So as I was showing earlier, there are things like a Purkinje cell, and you don't have one Purkinje cell, but the reason that it's named is that there's a lot of neurons that have this structure and kind of have a similar function or a basic kind of computational role within the brain. And what we find is, and this is in the case of the fly, that we can look at different cell classes within the fly, and we find that each cell class has its own log normal distribution, implying that there's not just both a fundamental generative process or developmental process that leads to the connectivity of the brain, but there's also one that kind of, or this can then be kind of narrowed down to develop individual cell types, which are then kind of the foundational units of computation within the brain. So you can think of this, I mean, not a perfect analogy, but if you look at like convolutional neural networks or different modules of connectivity within artificial neural networks, this is a way that the brain has its ability to generate these kind of modules of connectivity in a very reproducible way. So the final part of the talk is, why do we even care? Are log normal networks any different from the distributions that we generally look at? And obviously, we wouldn't be giving this talk if it wasn't, but we find that the log normal networks kind of slot in between this random network expectation and the scale-free expectation. And for those interested in these various network metrics, these are kind of how these network metrics can be derived from the parameters that define these distributions. And you find that, the log normal has these kind of exponential setups that are somewhat between just the linear scaling of k often and the kind of the scale-free or power law scaling that you see in scale-free networks, right? But just to bring this back down to values that we might be interested in, one of the things that we're interested in looking at is a correlation between brain size, because there's a huge variance in the number of cells in, let's say, C. elegans in a human, and the total degree or the max degree that we would expect within these systems, right? And we find that this Poisson or random expectation has this kind of log n scaling. The log normal distribution has something like is a very kind of this exponential and root 2 log n scaling. It's a little bit more complex, but it scales a lot slower at large values than that of the scale-free network. And these Purkinje cells, which I brought up at various points within these talks, that we saw that this huge input structure and are the basis of our motor function and exist in our cerebellum, we know that they're kind of famous for having 200,000 synapses, which is a very large value within the brain. There are these superconnectors, and they make 7,000 or almost 8,000 partners. So in a scale-free human brain, where you have 10 to the 11th neurons within these brain, you would actually predict that there's nearly 10 million partners that a cell like this would have, right? But the actual value is much lower. By comparison, in a log normal connectome, the value of the degree of a cell like this is closer to 1,000, which is much closer, both in magnitude and in value, to the observed Purkinje cells, which are a little bit of an extreme within the brain. So it provides kind of a better match actually to the data sets that we observe and the Kmax that are observed within these systems. And it also provides kind of a better understanding of various number of processes that we'll often study in network science. So for instance, looking at network robustness and the percolation threshold, which is related to kind of these knockouts within the brain, as well as spreading processes for the brain, it's very important as to how activity spreads within the brain. We want to know these values. And then finally, synchronization. How does activity in the brain synchronize and how is it passed out, which through waves and many other metrics is also well studied within the brain. And across all of these, what's an important factor is that the degree heterogeneity controls the network's functional and dynamical behavior, right? So this is a value that we also want to look at. And again, we find something very interesting, that robustness and synchronization for scale-free networks changes with that. So it's highly dependent on the value of neurons within each of these brains or within a network system. However, within a log normal network, you have this value, this normalized value, is independent of the number of neurons and only depends on the scale parameters that we've looked at before. This means that the robustness and the synchronizability of the connectome does not depend on the size of the brain. And in fact, there are very similar properties, or we'd expect there to be very similar properties across all of these systems, which is also important because all of these systems provide very similar functions. They keep an organism alive and they keep their organism interacting with the world. So it's important that this doesn't fundamentally change as you increase the number of neurons, but perhaps as one of the solutions might have been, is that you create local regions of connectivity, such as brain areas, that allows you to kind of create multiple pockets of computation and lead to more complex behaviors, as we might see within the humans or mouse. So just to summarize and to leave time for questions, we find that the degree distribution, the synapse distribution, and the length distribution are best approximated by log-normal distributions. We find that if we look at the connectome as a physical network, this allows us to analytically derive where the degree distribution and the synapse distribution come from. It allows us to also look at the origin of multiplicative processes responsible for the log-normal length distribution, which I didn't go into in depth, but is basically related to neurons being branching trees and how that leads to a multiplicative process that leads to a log-normal. And it also allows us to have these falsifiable dependencies between the parameters characterizing neurons. We also had some comments in the rich club behavior that, depending on the questions, I can go into. But overall, what it does is it represents a novel network class with very unique synchronizability, robustness, and spreading properties that aren't related to the network size, but are mostly related to the heterogeneity of the neuronal characteristics or the types of neurons, and therefore these scaling parameters that we observe. And as a kind of more hopeful or forward-looking message, we are our connectomes. These data sets are the future for both kind of understanding both who we are, as well as the diseases that occur during development or kind of later in life of the brain, and offer us the basis to start looking at how we can perhaps make ourselves more robust to these very detrimental diseases that we might observe within the brain. And kind of as a final note related to this, there's so many open questions that we're only getting to understand right now. So the talk that I kind of presented in the last 30, 40 minutes, it was kind of our attempt to try to answer these questions that we had down here. But we ran into two major issues. The first was in the quality of the data. We had to spend a lot of time actually cleaning these data sets and making them kind of accessible and being able to put them into a comparative framework. And the second was that while we wanted to move past the degree distribution, we found that the degree distribution and the kind of the basic network properties of these systems are so different from previous expectations, and that we didn't have models for them, that we focused on creating this baseline from which we can then build future network studies. And I think a lot of these future network studies, what will be important is being able to kind of analyze the physicality of these systems and better utilize this physicality in order to understand how local computations are being made, especially as we get connectomics in the next few years of larger and larger systems. So finally, I just wanted to have some acknowledgements. Ben led the study. He was the one who did most of the coding work, and we're co-first authors on this, but Ben was by far the lead on this. Andre is also on this paper. He's a neuroscientist that I worked with in order to get the data to a good state, to kind of understand some of the nuances of these different data sets and how we should be analyzing them, and what information to trust within these connectomes and what not to. Julia did basically all the theoretical background related to log normals, and we have really nice derivations, especially related to growth processes that she led. And Laszlo led the study and the writing of this network, of the manuscript that we're hope to release soon. This is my contact information. I'm also on Blue Sky. But I don't have a nice icon for that yet, and I'm funded by the Schmidt Center at the Broad Institute. So thank you.
Thanks a lot, Daniel. Okay, we can start with questions if there are people online. I see, Christina, did you want to ask a question? I see you.
Yeah, I've unmuted myself. Thank you very much. It's really interesting talk, quite fascinating, and great that you have the data. Though I've got a bad education in statistics, so I always tend to ask these questions. So I've got two questions. So the first one is, when you show your curves where you say it's a good fit, a statistician might think, that's a goodness of fit test here, right? The Kolmogorov Smirnov or something like that. Does this really fit? So that's
Uh, yeah. Sorry, I was showing that down here. So we did KS tests, and we found that
yeah,
log normals are generally the best fit. Again, there's a few exceptions, but across all data sets, like they're the best fit. And then we also do other fit tests as well,
yeah,
and we find, yeah.
sorry, but showing the statistic doesn't really mean that it fits, right? So usually there's something like a 5% level. I agree that, among the models that we tested, Lognormal
Okay.
has the smallest KS, but no, it's but it's and it seems that, well, I can't do the KS thresholds in my brain so quickly. I'm sure some of these will be significant, but some of these also might not be significant at the 5% level if you carry out a
Um,
KS test.
yes, I understand what you're saying. We have those results in the SI, but I think between the Zoom and kind of having two monitors, I can't pull them up. But
Okay.
I'll make a note of that. That if we don't discuss that yet, then that's something that we should spend more time
And
on.
and and another question is, so quite a nasty brain disease, if you like, is motor neuron disease. So, but there you've got these really long neurons, right?
Mm.
That could be up to a meter or two, well, longer
Mm.
than a meter, really. And that's probably not what your experimental setup is ready to do, or is it?
No. So for the mouse and for the human and for most, so there's a scale limitation of roughly 1 cubic millimeter of how big these reconstructions could be. So you can also use other methods to get lower quality data, and then you could trace kind of longer segments, or even these longer neurons as you're discussing. And we also looked at some of that data, and they also kind of fit a log normal distribution to the best of our understanding. But that's yeah, that we can't get that far given the current technique.
Yeah, but I mean, if it's the synapses length that drives everything, and if the synapses length is somewhat truncated by the experimental setup, that might also explain why you see log normal and not power law, because you don't
Um,
see the really, really, really long ones.
so we, I guess there's two things about this. The first is that in the cases that we saw that it was power law, we actually went back and looked at the data and realized that there were a lot of low quality neurons that were being released. And we did tests like this where we downloaded previous versions, or we actually took, in the case of the fly brain, and then started to kind of squeeze down the region that we're analyzing and cut off neurons within it. And we found that that pushes us away from the log normal, and kind of towards a power law regime. But also, what we did is that even in those cases, when we only focused on neurons that were fully within the volume, but all the partners that they actually connect to, we found that the log normal was still there. So that's kind of analysis one. And across some of these data sets, we actually have the full organisms or a full brain. So it's a very high confidence level as to whether we're looking at all the cells. The other thing that we looked at is for the mouse. As I mentioned, there are these tracer studies where you can inject within a certain region, and you don't get a lot of the local connectivity, but you get these long range projections, which, as you mentioned, we're cutting off. But these are still long range connections just within the brain. And we found that those are also log normally distributed. So, and we've talked to a lot of people. So there's someone connecting a full zebrafish brain, and they also were like, oh, we get really weird distributions. And I was like, well, do you have bad neurons within them? And actually, the moment that they cleaned up their data and they only had fully reconstructed neurons, they got a log normal. And this is a full brain of a zebrafish. So it seems that actually everything is once you get the good data, it trends towards the log normal rather than away from it through everything that we've tried.
Oh, that's amazing.
Yeah, thanks. Good to hear.
I have one also. So I was wondering, so you said at the end that this represents kind of a novel class of networks, these log normally distributed networks. Have you checked so far in any of the other like physical systems. So, I mean, like another biological one would be like fungi, for example. Like there was that cool stuff about how fungi recreates the Tokyo transit network or whatever. And then, so I guess
Yeah.
we do have some of those kinds of networks, but then also like other physical systems, like engineered systems, for example, that do have physical constraints, but very different physical constraints than the brain and
Mm.
neurons and synapses have. Have you or have you seen people checking specifically for log normal distributions in these physical systems when you're preparing the paper?
Yeah, well, we didn't ourselves, but there's been a big debate within the field related to this. I think the kind of the most famous or the biggest flare up on Twitter was related to Aaron Closet, where he took a lot of networks and fit log normals to them and found that they were a pretty good fit, perhaps related to power laws, and was challenging the sort of dominance of power laws. And I mean, I don't want to specifically get into that debate, but I think there's been a lot of discussion as to whether this is the correct fit. I think in my mind, it's more related to what is actually the developmental or the kind of the growth process that leads to a network that's a log normal. And while there's a lot of papers that have looked at whether log normals are good fits, in our cases, we can start to explain why we're actually seeing log normals. And again, I think you also mentioned like these fungi or these growth processes. We do know, I mean, just kind of as a brief initialization, right? Each neuron in our case is an incomplete tree, right? And a lot of these fungi are also kind of these branching processes or decaying branching processes that you might look at, right? And this Galton-Watson process, this is actually reversed on this slide, which was related to the I think it was like the like lineage, like royal lineages or something like this used to explain this, showed that branching processes with this kind of like probability of survival, right? Like trees that can end at different levels do lead, they're a multiplicative process that leads to log normal distribution. So there's a good understanding of this. But that was only, I guess, for kind of length or related to kind of like area or something like this within these systems. So it still was a jump in our case. And it took a lot of work to say, how do you get from something that explains your length to something that explains your other network properties that you might be studying?
Yeah. Cool. That's interesting. More questions? Yeah, go ahead.
Thanks for the report. Can I ask? So you show us in the beginning a nice video on how to construct the brain network with this 3D structure, and somehow the measure for the node strength is like a one-dimensional measure because it's rho times L. Since the dimension really matters in the physical system, so I wonder, is there a reason to choose that form?
So your question is related to the fact that they're a three-dimensional, like these are three-dimensional objects, as I was showing here, but we're only using a one-dimensional or metric.
Yeah, somehow it's got flattened.
Yeah. Well, I guess in part is that so that three-dimensional structure determines its reach, and part of, let me see if I can pull up this video, is that these neurons are extremely densely packed. So I mean, this is what the fly brain looks like. Is that I think this is also like only like 10% of the cells being visualized, right? So they have this massive 3D architecture, but part of what's going on is, and what we're seeing out here on the outside of it, is that all the cell bodies in the fly are on the outside of the brain, and then they all have to reach in, and there's no cell body or very few cell bodies on the inside of the brain. So let me see if I can kind of do this quickly, but like, yeah, so we see this kind of fly brain rotating, and you see that all the connections are on the inside. So even though it's a kind of their 3D structures, a lot of these 3D structures are related to kind of are related to spatial packing within the system. And therefore, it's more like it's really only along these line arbors that they have that they can make synapses, and the synapses are kind of points along them. So that's why it sort of works out that that this dimensionally reduced metric works. But actually, like how this partner choice occurs and how they actually select who to connect to, and of the cells that they're touching, which ones they actually connect to is something that we're very interested in. So I think this physicality is going to be much more important related to the specificity of the system and its ability to construct these modules of connectivity or modules of computation, which is not something that we went to in depth at this point, but is kind of like the next study that we want to hit.
Hey, Daniel, I have a question as well. Thank you very much for the nice talk. I have one confusion in my head, which is that you explain these networks as a branching process, but there's a lot of recurrence in the brain as well. So it may not be that locally the brain looks tree-like, which is what the branching process assumes, but there might be very short loops in the brain as well. And kind of a related question is that in your abstract, you mentioned that the 3D nature, the physicality of this network is really important. And because there is so little space in the brain, loops are maybe preferentially formed. Do you think this is accounted for in your model, and how?
Okay, so I think, I mean, I didn't want to go into this in depth because it's like, again, probably like another 15 minutes, but what we're actually saying is that each neuron is a tree rather than the whole network is a tree. So what we said that if you find a log normal distribution over the length of neurons, then from there you can derive the number of synapses and the degrees that it has. However, so all we wanted to get to is to say that why do we find neuron lengths that are log normally distributed? And for that, we said that each individual cell is a tree, right? So this loopiness is not something that we're accounting for here, but it's something that actually can arise from then the synapse burden that we see. And actually, this gets to kind of a fun point, which I didn't talk about before, is that we looked at this scaling, the sublinear scaling between degree and the number of synapses, right? So and one of the things that people love to look at within brain networks is the rich club phenomenon, which is to basically to say that superconnectors connect to superconnectors, and there's this rich club of highly connected and cells that have a lot of partners generally. And we find that actually this 3D structure and the kind of the size of these cells is related to this, is that the cells have so many synapses. And when you try to randomize the system, which is what you would do with a rich club coefficient or to kind of calculate rich club coefficient, if you do this at the synaptic level, have to kind of not just disconnect like the degree values, but the kind of individual locations where cells can connect. The superconnectors have so many synapses that they can only connect to superconnectors to fill those values, right? Like they're more probable to do this. So we found that like this 3D information, for instance, is very important for the rich club coefficient, and basically says that any brain network will somewhat trivially will be a rich club because it's the only way to actually fill these synapse numbers in a randomization setting. So the 3D-ness might be something that we might need to take a little bit out of the abstract, given some of these claims, because we mostly just look at this kind of this tree structure, but the kind of the 3D information, but I admit is like not fully accounted for in some of our studies.
Thanks.
Okay.
Great. Any more questions in the room? If not, anybody online, if you want to unmute or raise your hand, last chance. Seeing none, then thanks again, Daniel.
Thank you. And I'll just leave this up for the next few minutes. If anyone wants to reach out, then please do, because we'd love to hear more.
Great. Yeah. Thanks a lot.
Thanks.